Okay, so it is my pleasure to introduce today our next speaker for the Math Hub seminar, Krishna Pusuluri. He's a postdoc with Dr. Vince Cahot at Trent Torres Center, Georgia State University. Prior to this, he worked as a postdoc at the Mori University with Dr. Astrid Prince and obtained his PhD in neuroscience from GSU with Dr. Andrei Shilnikov. He also has a background in computer science and worked as a software engineer with Yahoo and as a research at India School of Business. Christian's research interests span complex systems, chaotic dynamics, modeling neurons and neuron circuits, multistable center pattern generators, neuroimaging, analysis, and machine learning. And without further ado, please, Krishna, thank you very much, and you may begin when you're ready.
All right. Thank you so much. Thank you for having me here. I hope you can hear me well. Okay. So in today's talk, it's about how we leverage parallel computations to study complex dynamics arising in neuroscience and other systems from physics with chaotic dynamics and so on. So most of this work was my graduate research with Dr. Shilnikov, and this is kind of like a continuation from his talk 2 weeks ago. So this is how this talk is organized. First, I'll give a very brief overview of various parallel and hybrid computing approaches. So if some of you are new to this discipline and want to use parallel computations in your work, I'll just give you some pointers as to how to get started. Then the rest of the talk is going to be a broad showcase of how we use these technologies to study complex dynamics in a variety of different systems. So the first part is a brief overview, like I said, of parallel and hybrid computing approaches. So you all must have heard about CUDA and how you can use that to run your code on GPUs. So CUDA is more of a low-level programming language, similar to C, with some special directives to say how to run different things on the GPUs versus on what's to be run on the CPU, and how you move data between GPU memory and CPU memory. So CUDA is a low-level programming system, and may not be the best place to begin learning parallel computing. So the other alternatives that I found really useful are OpenAcc, OpenMP, and OpenMPI, which are directive-based approaches. So it's very simple to use these approaches. So if you have like shown in the code segment here, this is a loop where we are computing a long trace for different initial states, and you're running, let's say, millions of these trajectories for a million different initial states. And if you run this on a CPU, it will take a very long time. So if you are instead using a directive-based approach like OpenAcc, which can help you run this code on a GPU. So basically, you'll add a small directive, hash, pragma, ICC kernels, saying that this is the loop you want to parallelize on the GPU. And then this compute traces method, you're saying here that it's a routine that you want to run on the GPU threads. So, and then there are a few other mechanisms by which you can move data between the CPU and GPU memories. And so OpenAcc will help you parallelize this code for the GPU. So you can use the same C code. You can tell the compiler to run it as a straightforward linear version of the code, or the same code you can tell the compiler to use the OpenAcc directives and run on a parallel processing GPU. So OpenAcc can help with this directive-based approach for parallelization on GPUs. OpenMP is probably the best place to start learning to parallelize your code. It just does this on parallel CPUs. So let's say you have an 8-core CPU, and you just want to run this loop parallelly on 8 different processors. So you just add these directives, OpenMP directives, and you can use GCC compiler with OpenACC, with OpenMP, and parallelize this code on the CPUs. And MPI is when you want different threads of your loop to interact with each other. So OpenACC and OpenMP you can use when your threads are independent of each other, and you can just run them in parallel. If you have more interactions between the threads and you want to send different messages from one thread to another depending on different conditions, then you start using MPI, which stands for message passing interface. So this is just showing how you can scale your performance with these different parallel approaches. You can achieve orders of magnitude performance improvement once you parallelize your code to run on GPUs. So I think, so this is the blue on the top is the serial version of the code, and at the very bottom, it's parallelized with OpenACC and then MPI to communicate between multiple GPUs. Then you see a couple of orders of performance improvement. And very quickly, these are some resources that you can tap into to learn about all of these techniques. I personally learned from these HPC workshops that are conducted by Pittsburgh, and they have multiple satellite sites. So they used to conduct these courses at Georgia State and Georgia Tech. I see there is one coming up for OpenACC. That's on November 7th. I think you should be able to register online. And this is an OpenACC workshop. So it runs at Georgia Tech also. So I saw, usually at Georgia State, we had multiple machines. So you go in person, sit there and go through this workshop. And NVIDIA GPU TechConf has a lot of resources that you can tap into, and then on-demand videos that you can use. And these are the different compilers. They're all free that you can download and use. So basically, this is how you tell the GCC compiler that this is that you want to use OpenMP parallelization across CPUs. And if you do not give this directive, then it will just run as a serial code. It's very simple to use OpenMP and OpenACC, and you should all just try it in your code. So now, so next I'm going to demonstrate how we use these parallel computing approaches in our own research. The first topic will be multistable neural networks and central pattern generators. So large brain networks and large central pattern generators are composed of smaller structural and functional building blocks called as motifs. Now, this is a study looking at monkey cortices, and they found these different smaller sized motifs that are expressed in large numbers. And the networks on the right, these are central pattern generators that control the swimming behavior of two sea slugs that are extensively studied by our collaborators, Akira and Paul Katz. So even these CPGs are composed of smaller building blocks, half-center oscillators, which are, if you see two on the left and two on the right, these mutually inhibit each other. So these dotted lines represent inhibitory connection between these two cells. And then there is electrical coupling, there is excitatory coupling. So these smaller motifs of half-center oscillators are coupled together with different excitatory, inhibitory, and electrical coupling. So by studying these smaller networks, we can understand how rhythmic behaviors arise in larger networks and how they can give rise to robust and resilient rhythmic patterns. So this is something that Dr. Shilnikov talked about 2 weeks ago. So this is looking at a 3-cell central pattern generator where each of the cells mutually inhibit each other. So cell 1 and 2 inhibit each other, and as does the cell 3 with each of the other cells. And these are looking at stable rhythms that are produced by this circuit. So this rhythm on the top is a pacemaker rhythm, where when the blue cell fires, it suppresses the other two cells. And then when the blue cell goes down, the other two cells become active. So one cell is up, the other two cells are suppressed. And by symmetry, there can be 3 such pacemaker rhythms. And the second rhythm is a traveling wave rhythm, where the first cell fires, then the second and the third, and then that keeps repeating. So we are using Poincare return maps. So this is showing the phase plane for the voltage variable and the recovery variable. So there is the cubic nullcline for the voltage variable and a slow nullcline. And together they produce this oscillatory behavior. And on the right, this is a Poincare return map and a sweep across different initial conditions. So we start from different initial conditions. So on x-axis is the phase lag between cells 1 and 2. As you vary that phase lag from, let's say, both cells 1 and 2 are starting in phase, or versus when it's 0.5, they're out of phase. Okay. Similarly, on y-axis, you're showing the phase difference between cells 1 and 3. So when we run these trajectories, starting from these different initial phase lags for the 3 cells, we find these 5 different regions. So pink and black correspond to the traveling wave regimes. So one is the clockwise traveling wave and the other is the anti-clockwise traveling wave. And the blue, red, and green regions correspond to pacemakers, as shown in this top panel. Okay, so the white dots correspond to the fixed points or the attractors for these regions. So we employ these Poincare return maps to study how multistable, what sort of multistable rhythms can come up in these networks. And as you vary different parameters, such as the synaptic connectivity or the intrinsic parameters of the cells, how do these multistable behaviors change? How can you get rid of pacemakers, and how can the circuit produce just traveling waves? How can it produce just a single traveling wave, a clockwise traveling wave, and not the other one? So, so these Poincare maps are really useful to study multistable behaviors. And on the right, it's just showing how you can vary the synaptic connectivity just by strengthening one of these inhibitory connections. You can initially get rid of all these 5 different stable rhythms, and you just have a wave-like pattern with phase slipping. And so there's a stable phase slipping pattern, and there's an unstable phase slipping pattern. And then you eventually can get rid of all these rhythms to give rise to an ergodic torus, where there are no fixed points, no stable cycles. Okay, so the Poincare return maps have been used to study 3-cell dynamics. Now imagine you have one more cell. Then you'll be so for 2 cells, you have, for 3 cells, you have 2 phase differences, and you can plot it on a 2D map. If you have 4 cells, you have 3 phase differences, and it's already a 3D system. And if you have more and more cells, it's not easy to visualize and look at these different basins of attraction. So what we are looking at here is homogeneous 4-cell configurations. So these are different homogeneous 4-cell network configurations with mutual inhibition. So this is a one-way loop where one cell is inhibiting the next, and so on, and there's a circular loop. And this is again a 2-way loop, and this is more of a mixed connections, and this is the fully connected network. And as I'll demonstrate shortly, so this network at the very end is a robust multi-stable network, while the first network is a bistable network. And the networks B and C are produce robust monostable rhythms. This is how we can study the effect of network topology on the kind of rhythms that are produced by these networks. So how do we study these 4-cell networks using Poincare maps? So for a 4-cell network, as I mentioned before, so there are 3 phase lags that you're looking at, and it will be a 3D phase space. And it cannot be visualized so easily, and it's an exponentially increasing algorithmic complexity, because you're looking at, say, 25 different phase initial conditions on each axis. So it's 25 power raised to the power of the number of neurons, -1. So what we do is instead we use clustering algorithm. It's a hierarchical clustering algorithm, and you measure the circular means of those clusters. So all the blue regions, we cluster them together where they're converging and compute the cluster mean. And we also compute the cluster standard deviation to measure the variability within that network's rhythm. So using this, what we can identify is this 4-cell network can produce all these different rhythms. So this 4-cell network can produce this paired half-center oscillator rhythm, where 2 of the cells are active and they suppress the other 2 cells. And by symmetry, you have 3 such paired half-center rhythms. Or you can have a pacemaker rhythm where 1 cell is active, as shown here. 1 cell is active and it suppresses the other 3. And when this cell goes down, the other 3 become active. And this keeps repeating. And there are 4 such pacemaker rhythms. And you can also have a synchronized state in this network, even though this is a mutually inhibitory network. It can still produce a stable synchronized state. And you can have a full traveling wave rhythm where the first cell fires, then the second, then the third, then the fourth. And there are 6 such traveling wave rhythms by symmetry. And there's a mixed traveling wave rhythm where 2 of the cells are firing in phase. And then there is a traveling wave with the third and the fourth. And there are 12 such mixed traveling wave rhythms. And what this parametric sweep is showing is as we vary the synaptic connectivity, which is these Gs, the strength of the synaptic connectivity across this network. That's on the x-axis, and on the y-axis is the externally applied injected current. And as we are varying these on a grid, on a 7x6 grid, we are using this clustering algorithm to run the Poincare 3D Poincare phase space analysis, and then the hierarchical clustering algorithm to identify which rhythms exist for those parameters. So, for example, in this parameter, for these set of parameters, you have this yellow region, which is the 3 paired half-center rhythms, and then the green region, which is the synchronized state. So for this set of parameters, the network can produce multistable behaviors with half-center oscillator, paired half-centers, and a synchronized state. And in this region, you can have paired half-centers and the fully traveling waves simultaneously. So different initial conditions will produce stable rhythms that could be paired half-centers or fully traveling wave. So this shows some of these rhythms. So this is a fully traveling, a full traveling wave. So one cell is becoming active, then the next one, then the next one, then the next one. So that's within these pink regions. Then this is a paired half-center where 2 of the cells are firing together, and then the next 2, and so on. And this is the fully synchronized state where all 4 cells are eventually converging together, even though there is mutual inhibition between each of them. So here we are looking at how network transitions can be studied. So how can you transition from a network that is that produces bistability to a network that is robustly monostable? So here we are increasing the anti-clockwise inhibitory loop, the strength of the anti-clockwise inhibitory loop in this network. So eventually it becomes this network on the right. So this network is initially bistable with the paired half-center and the fully traveling wave rhythms. But as you see, as the strength of the anti-clockwise loop increases, it becomes robustly monostable with just a single paired half-center rhythm. And here we are transitioning from this bistable network to a robust monostable network with that produces a full traveling wave. And here we are traveling from one monostable network on the left to another monostable network on the right. So these are the paired half-center rhythms and the full traveling wave rhythms. Yes. In C? Okay, so in B, basically we are adding this cross inhibition. So the network on the left, basically whatever is on the left and what's compare it with the network on the right. As you add the missing connections or you strengthen and weaken to transition this network to the next, you see the gradual transition of the rhythm that network produces. So that could like kind of explain how different CPGs could evolve to produce one monostable rhythm. I'll show some of those CPGs in the next section where we are talking about the swim CPGs that I showed before. It produces one monostable rhythm. Yes.
So I'm confused why a 4-vertex network can support synchronized states, but a 3 seems unable to. Really need to connect to why that's 3.
I mean, I can be absolutely sure, but my guess is it may be possible in the 3-cell network as well. But the regime may be too small. Even in the 4-cell network, it's a very small regime in the parameter space that it produces this stable network. Right. So
Is this like try this on like 5 networks?
no, I haven't looked at like larger networks. This study is mostly looking at like the small 4-cell networks. We moved from the 3-cell to the 4-cell. But I think it would exist, but within a very, very small parameter regime.
Parity question, or if it's like a concentration type of function.
Yeah,
Like,
yeah.
as you get bigger networks.
Okay, so next I'm going to talk about symbolic methods and how we can use these symbolic methods to study various rhythms, various activities or dynamics produced by isolated neurons, as well as dedicated CPG circuits. So neurons can produce different behaviors, such as they could be tonic spiking, or they could produce a bursting rhythm, or they could be quiescent, or they can also produce a complex chaotic bursting with mixed mode oscillations like shown here. So this is a trajectory from plant model, and where within a burst, the number of spikes is unpredictable. And then successive bursts are separated by an unpredictable number of subthreshold oscillations. So this is a chaotic mixed mode oscillations trajectory. So we are, in order to study these neuron models and even neural circuits, we are employing a symbolic encoding scheme. So it's a very simple scheme. You could use partitions in voltage or partitions in time. So here we are using 3 partitions. So anything below -60 millivolts is marked by a different symbol, and -60 to -40 is marked by a symbol B, and then C, and then D. Okay. So basically we are looking at events of maxima or minima in this voltage trajectory. And then as shown in the panel on the right, so we are looking at events of the red dots are the events of maxima or minima. And you could use a voltage partition. So then this point here would correspond to the symbol D. This point here would correspond to the symbol B. Or you could use a partition in time intervals. And let's say we have a 100 millisecond partition. So any gap, any interval that's less than 100 milliseconds is given the symbol A. Any gap that's greater than 100 milliseconds is given the symbol B. And using this kind of symbolic representation, we are basically converting this voltage trajectory into a sequence of symbols. So if we are using just the voltage partitions, this trajectory would be represented by the symbols D, B, D, B. And if it's a tonic spiking trajectory, it's a periodic sequence D, B, D, B. Similarly, if you are using time interval partitions, this would convert into capital B, capital A, capital B, capital A, and so on. And if it's tonic spiking, it's a B, A periodic sequence. And you could also use a combination of voltage and time interval partitions. And then what we are doing is we are looking at long trajectories and looking at whether this trajectory is periodic using a circular hash function. And that identifies whether there's periodicity in the sequence, which would correspond to tonic spiking or bursting. And if the sequence is aperiodic, it means there is a chaotic, something like a chaotic mixed mode oscillations trajectory. And such trajectories we process with complexity algorithm called LZ complexity. It basically creates a dictionary and looks at whether, as you add a new symbol, is it already existing in the dictionary or is it creating a new word? So it increases the complexity of the dictionary. So using this symbolic approach, so here, we are looking at a 3D leech heart interneuron model. It can produce a bursting trajectory or a tonic spiking trajectory, and this is the 3D phase space, and this is the map. So this is the parametric sweep on the right. As you vary to biophysical parameters, IAP is the externally applied current, and VK2 shift is the activation for the potassium current. And as these parameters are varied, this model can produce, in the white regions, it can produce quiescent behavior, or in the gray regions, it's a tonic spiking trajectory, like this pink trajectory here. And within these colored regions, it produces bursting. And as you move from one band to the next, it's spike adding, bursting with spike addition. And at the boundaries, it produces chaotic spiking. And these we identify with the LG complexity algorithm. And here I'm showing 4 other models. So this is a 3D Hindmarsh-Rose model. Again, this is in this region, it produces bursting with spike adding. And here it's tonic spiking. And at these boundaries in the gray regions, it produces chaotic spiking. And again, this is for a 12-dimensional hair cell model. So now, as I showed before, so we can use this symbolic approach and then parametric sweeps. So basically, how do you obtain this parametric sweep? So for each set of parameters on the x-axis and the y-axis, we compute a long trajectory and then process that trajectory using this symbolic algorithm and look at the long-term behavior and basically assign a color to each different behaviors. So then, so basically, if you are running a 1,000 by 1,000 grid, you're computing 1,000,000 trajectories to obtain the sweep. So if you do this on a CPU, it takes a very long time. So we parallelize on the GPU using, most of this is with CUDA, and but you can use all the other approaches as well that I mentioned. Now, I'm going to show how we can extend the same approach to a network, a small network. First, I'll talk about the single neuron model. So this is a 6-dimensional plant model. It was originally built for the sea slug Aplysia, and it's an intrinsic burster. So then Denise, who was a previous graduate student from the lab, she extended this model to study the swim CPG in the sea slug Melibe. So she added a parameter x shift, which modifies the dynamics of the slow variable, and it can eliminate bursting in this model. And this parameter calcium shift controls the reversal potential of calcium, which cannot be exactly determined in these cells. So for this sea slug, these neurons are either intrinsically tonic spiking or quiescent. So they do not produce bursting behavior intrinsically. So bursting is an emergent property of the network. So the region of interest for this model, in order to apply it to this particular swim CPG, is within this boundary between the tonic spiking and quiescent regions. But as you see from the sweep, this model can produce other behaviors with bursting with spike adding within these different colored regions. Pink region is tonic spiking, white region is quiescent. And at the boundary between tonic spiking and bursting, which is magnified here, it produces chaotic spiking trajectory as something that's shown here. So it produces a chaotic number of spikes. Whereas at the boundary between bursting and quiescence, this small black region is magnified here. So white region is quiescent, these colored regions are bursting, and within this boundary, it produces this chaotic mixed mode bursting trajectory. So within the burst, there are a chaotic number of an unpredictable number of spikes, and between successive bursts, there is an unpredictable number of subthreshold oscillations. And this is just showing this in this 3D phase space. Yeah.
How do you quantify the transition into these from the symbolic level? Is it just like
Okay.
you look at the running time average, see if it
So you convert the trajectory to sequence of symbols, right? And then detect whether there's periodicity within that sequence.
Right.
Okay. So if it's just tonic spiking, you have two symbols, the maxima and minima, and it keeps repeating. So, and at different parameters, if they converge to the same symbolic sequence, they're assigned the same color. So within this pink region here, all those trajectories are converging to the same symbolic sequence.
So chaos in your quantification here is just saying that there are no periodic subsequences.
There are no periodic subsequences. And then LZ complexity, what it tells is the complexity of the symbolic string. So whether it is more chaotic or less chaotic, something like that.
Okay.
So that's determined by whether it's dark gray or light gray that's shown here. So again, what I showed here is for a single neuron, this parametric sweep, how this model can produce different behaviors. And this is the region of interest because these cells in isolation are either tonic spiking or quiescent. Okay, so now how do you extend the symbolic scheme to study a simple network like a half-center oscillator? So these two cells are mutually inhibiting each other. So again, you use a partition in voltage. So anything above this purple line is, okay, so we are only looking at events above this purple line. And then we are also using a time partition of 2.5 seconds. So if the time interval between successive events is greater than that 2.5 seconds, we mark it by the symbol B. If it's less than that 2.5 seconds, you mark it by the symbol A. So that tells you whether it's an interburst interval or interspike interval. Okay, and then you use different symbols for cell 1 versus cell 2. And if it's bursting behavior, where the cell 1 bursts and then the cell 2 bursts, this symbolic encoding scheme would convert it into the string DCBA, or alternatively, it could be BADC. The circular permutations are all. And again, we use circular hashing to determine periodic sequences, Lempel-Ziv complexity to determine bursting behaviors as we vary different parameters and compute these trajectories. And here we are looking at these two cells and how bursting emerges in this half-center oscillator. So as you vary the parameters, so these are alpha and beta are the parameters controlling the slow synaptic dynamics for the mutual inhibition. Alpha, increasing alpha implies a stronger synapse. Beta is the rate of removal of the neurotransmitter. So increasing beta implies a weaker synapse. So when these cells are intrinsically quiescent, C and D, panel C and D, the cells are intrinsically quiescent. So when alpha is 0, when the synapse doesn't exist, the cells are in this white region and they're quiescent. And as you increase the strength of the synapse, it gradually produces this blue region where the cells produce a bursting behavior, a half-center bursting behavior. The cell 1 bursts and then the cell 2 bursts, and then that keeps repeating. Okay, and the same can also arise within cell for panels A and B. The cells are intrinsically tonic spiking. So when alpha is 0, the cells are in the red region where both the cells are tonically spiking, as shown in panel A here. And as you increase the strength of the synapse, you have this blue region where you see the HCO burst. One cell is bursting, then the second, then the third, then the first, that again, and then the second. So it keeps repeating. If you strengthen the mutual inhibition further, then only one of the cells is tonically spiking and it suppresses the other. So that's this green region. And within near the transitions, you see this gray region where you have a chaotic bursting trajectory as shown in this panel B. Okay, so next, we are using the same approach to build in a bottom-up fashion a 4-cell CPG that controls the swimming behavior of the sea slug Dendronotus. So in Dendronotus, these neurons are not intrinsic bursters. So when isolated, they do not burst. They're either quiescent or tonic spiking. And then even the half-center oscillators do not produce bursting rhythms. So instead, they need the full circuit of the 4-cell network with all the mutual excitation, mutual inhibition, and electrical coupling to produce a network burst. So that's what we are trying to gradually build. This is showing it for a single neuron. The purple region is tonic spiking, white region is quiescent, and this model can produce bursting within the small region, but intrinsically, it's within this tonic spiking or quiescent region. Okay. Then this is showing for the half-center oscillator between cells 3 and 4 with mutual inhibition, where intrinsically they're tonic spikers. And as you increase the strength of the inhibition, it can produce suppressive rhythm. So only one cell is tonically spiking and suppressing the other. But it does not have the blue region as shown before, where there is the half-center bursting rhythm. So in order to get that half-center burst, you need this other half-center as well as the all the other connections. So this is the full 4-cell circuit. And here we are looking at the mutual inhibition between cells 3 and 4 on this axis, and the cross excitation from 1 to 4 and 2 to 3, that's on the y-axis. So when the mutual inhibition is very low, these are in the red region, which is the cells 3 and 4 are tonically spiking. And as you strengthen the mutual inhibition, they go to this green region where one cell suppresses the other, as shown in this panel. Okay. As you increase the cross excitation, then you start seeing this blue region where you have this full network rhythm. So this is the rhythm produced by the CPG in even in experimental recordings. Okay. So again, we are looking at how the balance between mutual inhibition, excitation, and gap junctions between these different neurons in the circuit control this bursting rhythm. And it, we are also looking at the number of spikes per burst and different other measures to match them with the experimentally seen behaviors in the circuit. So next, I'll quickly show some applications of these same techniques, symbolic methods, parametric sweeps, parallelization across GPUs to systems with chaotic dynamics from physics and nonlinear dynamics. So this approach with symbolic dynamics we call deterministic chaos prospector. It can be used to study Lorenz-like systems. They can identify various structural building blocks with homoclinic bifurcations, characteristic spirals, T-points, inclination flips. All these can be identified using the same approach. And they offer significant performance improvements compared to other existing techniques like Lyapunov exponents or parametric continuation. So this is a Lorenz system with the characteristic butterfly attractor. So as you start a trajectory, it produces this unpredictable movement between the left and right wings of this butterfly. And here we are looking at just one variable. And then when it goes to the positive wing of the butterfly, the right wing of the butterfly, that's shown in blue, and on the left wing, it's shown by green. And then you can convert this trajectory into a sequence of ones and zeros. So whenever it's on the right wing, you mark it by the symbol 1. And when it goes to the left wing, you mark it by the symbol 0. So then this complex looking trajectory is converted into a sequence of ones and zeros. And then we design a metric No, from the sequence of ones and zeros, you multiply it by powers of half. So from the sequence of ones and zeros, you get a metric and use that metric to construct these parametric sweeps. So these are two different parameters from this model. As you're varying, so take one of these panels, panel A, as you're varying these parameters, for each combination of parameters A and B, you compute this long trajectory, convert it into a p-value, and then use a color scheme. So two different trajectories that have the same topological behavior. They will produce the same p-value and will have the same color in the biparametric scan. So then the boundaries between different regions are the homoclinic bifurcation curves. So meaning that the trajectory is coming back to this fixed point. So here, panel A is showing the transient behavior, which will identify all these various homoclinic bifurcation curves and the characteristic spirals. And then on panel B, we are looking at long-term behavior. So you look at a very long trajectory and say, skip the first 1,000 symbols and look at the next 1,000, 5,000 symbols. And then again, use the same algorithms as before, detect whether the sequence is periodic. If it's periodic, it means that it's a stability window. If it's aperiodic, then we use LZ complexity, and it's a chaotic region where the behavior is unpredictable. So here I'm showing the same thing for a 6-dimensional nonlinear laser model. So if the trajectory goes to the right, as shown in blue, it will be marked by the symbol 1. So we are starting along the right unstable separatrix of this fixed point. So the first symbol is always 1 because it's going to the right. And then if the next turn it makes on the right, as shown in blue, it will be marked by another symbol 1. If the next turn is to the left, it will be marked by the symbol 0. So it will be 1, 0. At the boundary between these transitions, it will come back to this fixed point. And so it's a homoclinic bifurcation curve. And this is also showing various periodic orbits that this model can produce. And this gray trace is a chaotic trajectory again. And this is a heteroclinic connection where the trajectory is the red trajectory is going once to the right and then to the left, and then merges with this fixed point. And this is showing an inclination flip bifurcation. I won't go into a lot of details, but we are projecting beta, this variable, and D23, that's the 6th variable. Okay, so for this system, on the panel on the left shows the short-term dynamics with all these various homoclinic bifurcation curves, the characteristic spirals of Bikov T-points, and some of these are magnified. So this magnified region here, it shows a codimension 2 point called inclination flip, from where all these bifurcation curves seem to arise. Okay, and these are magnifications of 2 T-point spirals, where all these homoclinic bifurcation curves converge. And this panel here is the reconstruction of the same thing using continuation, parametric continuation. So one of our collaborators, Hill Mayer, he was the one of the creators of MatCon. So he tried to reproduce all of these bifurcation curves. So in order to reproduce this with parametric continuation, you need to manually reproduce each single curve. It's an elaborate process. And whereas this parametric sweep here with GPUs, you can produce it in just like 10, 20 seconds. And here we are looking at the long-term behavior of the system. Again, these different colored regions are stability windows. So it's periodic. It could be a single loop or a double loop and so on. And this gray regions are regions where the behavior is chaotic, with dark gray implying more complex behavior as opposed to light gray regions. This is again showing some of these inclination flip points that I mentioned. So in the short-term behavior, all these homoclinic bifurcation curves are arising near these inclination flip points. And in the long-term behavior, these stability windows also seem to arise from close to these inclination flip points. Okay, and so like I mentioned, to produce these 2D sweeps, you can do this in a matter of a few seconds. So now what we can do is we can look at three-dimensional bifurcation surfaces. So you do produce multiple 2D surfaces, multiple 2D sweeps, and construct these complex three-dimensional bifurcation surfaces to look at how this behavior is changing. So this was a figure from 1993 that's hand-drawn, showing how this is supposed to be near a parametric saddle in the 3D space. Here, it is actually reproduced using this GPU parallelization and symbolic method. And again, here, these are 2 T-points above and below this parametric saddle. And how is it embedded in the 3D space? We can study by constructing this detailed 3-dimensional bifurcation surface. And okay, I don't have this video. That link isn't working. So, but again, this is the 3-dimensional embedding of these of the saddle. It's like a bent scroll structure. So when you look at the 2D plane, the top 2D plane, you see these 2 spirals as 2 different spirals, but essentially they're connected to the through a 3-dimensional bent structure. And this is a different system. This is the 3D Rabinovich system. Again, we can look at different characteristic spirals, homoclinic bifurcation curves. And on the right, this is the 3D Rossler system, looking at the stability windows and gray regions are the chaotic regions. And these are called shrimps because they look like shrimps. Okay, so I think in summary, we are using these parallel GPU computing approaches and combining it with Poincare return maps, symbolic encoding schemes to study a variety of different systems from neuroscience and other nonlinear systems. And we can, for neural networks, we can look at what drives whether a network is monostable or multistable, and how do network topologies affect these rhythmic behaviors from the CPGs, and how intrinsic and extrinsic properties of the cells and networks, synapses, control their behaviors. And for other nonlinear systems, Lorenz-like systems with chaotic dynamics, we can use the same approach to study the bifurcation structures that underlie complex behaviors in these systems, and which could be used to develop other novel applications in biological and engineering systems. So these are some of the references for all these different projects, and all of the code is available. So you are free to just download and try it out, and you can reproduce all of those figures if you have a GPU or even on CPU. And I would like to thank all the members from the Shilnikov lab. So they have been part of, we have all been part of different projects that I mentioned. So Denise, Jack, Jasam, these were instrumental in studying the CPG, the swim CPGs, and Jared, Eustace, Jeremy, and Drake, Denise and Tingley, they were all part of the multi-stability analysis, and Tingley and Jeremy were part of the other Lorenz-like systems. And some of this work was funded by NSF, and NVIDIA sponsored some of the GPUs that were used in the study. Thank you, and I can take questions now.
Thank you very much, Krishna. Let's thank the speaker. We have some time for questions, so if anyone online wants to ask a question, you can unmute yourself or type it in the chat, and I will communicate it to the speaker. Right now, go ahead.
Yeah, go ahead.
Can you go back to the Lorenz system? So I'm a little confused as to like what I'm just missing the point. Because so this coding only makes sense when you already know that you have like a span portion. You already know that you have all these component sort of connections, and you know that you're topologically conjugate to each thing. So I'm a little confused as to why you want to create such complicated figures to express the chaos that we need in order to make the figures make sense. So could you, because if you don't know already that you're conjugate to a horseshoe, right? Then you don't know already you're conjugate to a two-chip. This p-value means nothing, right? Am I like misunderstanding something?
So I guess I mean, let me know if I understand this correctly. So I guess the question is, what's the point of all these maps, right? Like the parametric maps, and unless you know the and can you use it without knowing the underlying behaviors of the system? Yeah. So I showed the symbolic methods for the neural systems first, and then showed it for these other systems. But the actual, the way we developed this was first we were using this for these Lorenz-like systems. And then we started applying it to neuron models without even knowing what's going on inside the system. So then you can use it to study different systems without knowing elaborately about the details of the model itself.
I guess I don't know. I kind of felt like because it seemed like you had like relatively specific wave spike cutoff points that were like clear from time series data. Like it was clear from time series data that there were some numbers that you could like say if you cross this, then you're in a spike, right? And so maybe that's not a full conjugacy, but at least that makes sense. But so in general, in order to apply these symbolic sequences, if you don't have chaos, if you don't already know that a span portion exists, and have this like coding conjugacy, is the idea that just as long as you have sort of a well-developed way to separate space into specific behaviors, then it is.
I mean, like some of the neuron models, the regions of chaotic bursting are very small. So you may not even see any chaotic bursting when you look at the parameter space, but you can separate out the trajectories. You can classify different trajectories, right? So based on how they're bursting or so I guess my point is that you can use the symbolic encoding to classify different
but
parts of the space.
You know, it's a conjugacy. Like you're not saying anything necessarily about the dynamic system rigorously. You're just saying
yes. And I guess it depends upon your symbolic encoding scheme. You for the Lorenz systems, you use a different encoding scheme, and where there is some conjugacy, where in the other systems we are just looking at the spike numbers and not, let's say, the interspike intervals and other things, right? So you can adapt your symbolic encoding scheme to determine as much detail as you want to look at from the system. I think it depends on the system as well. So.
We'll stop the recording in case anyone is shy. We'll have time for a couple more questions.